Infectious diseases and immune response. Infectious diseases can be caused by many factors, and we're going to talk about all of them right here, and in addition, what the immune system does to get rid of them. And we'll finish up with some specifics on HIV, tuberculosis, and malaria in Malawi. Viruses. Viruses are intracellular parasites. They can only reproduce inside cells. Viruses are mostly removed by cells that kill the virus-infected targets. Innate immune response. The natural killer cells of the innate response kill virus-infected cells. Some viruses, however, might evade that early response if they get inside cells and begin to reproduce quickly. The adaptive immune response, that response with cytotoxic T cells that kill the virus-infected cells, may take 7 to 14 days to develop. The T cytotoxic cells work only with help from antigen-presenting cells, or known as APCs, and T helper cells. An effective adaptive immune response to viruses is usually dominated by these events. First, the APCs phagocytose, that's just a fancy word for ingest, the virus antigens and present pieces of the virus on their surfaces. The T helper cells recognize the viral antigens on the APCs as non-self and make lymphokines in response. T cytotoxic cells that are activated by those lymphokines, they then recognize the virus-infected cells and they will kill them. HIV, human immunodeficiency virus, is a retrovirus that is transmitted into the bloodstream by sexual activity, intravenous drug use, or from mother to child during pregnancy, labor, delivery, or breastfeeding. Almost anyone can be infected. It is an immunodeficiency virus because it causes deficiency in the immune system. All retroviruses carry their genetic material in the form of RNA, not DNA. So here's a picture of one of these HIV cells, and you can pause that for a second and check out the terms. HIV begins the infection when the HIV glycoproteins bind to CD4 proteins found on T helper cells. Fusion. The viral RNA enters a host cell. 3. Reverse transcription. A special enzyme, reverse transcriptase, copies the viral RNA into DNA. The next step within the CD4 cell, which is this yellow and blue color here, that's all the CD4 cell, the viral DNA integrates into the host DNA and sets up a long-term infection in there. 5. Replication. The viral DNA directs the host cell to replicate the HIV. 6. The assembly. New virus particles are made. And finally, 7, which is budding, the new virus particles bud off the host cell and infect neighboring T helper cells. Some viruses infect the host cells and then don't reproduce frequently. They quietly set up a persistent chronic infection by presenting very few targets for the immune system. The virus then reproduces at a low level and does not make the infected person ill. HIV is an example of a persistent virus, a virus that hides away from the immune system and infects for life. A person with an HIV infection appears healthy yet can infect others. Natural selection of persistent infection has ensured that this type of virus will spread before sickening the host. This is an ideal adaptation for a virus. 2 additional factors make HIV a deadly disease. 1, the T helper cell decline, and 2, HIV's high mutation rate. The cell that we showed on the previous slides was a T helper cell. So that's the first one here, the T helper cell decline. They are the main target for HIV, and T helper cells are the most important cell in the adaptive immune response. As HIV replicates inside the T helper cells and releases new virus particles, it kills the T helper cells. When T helper populations decline, adaptive immune responses decline, and patients can't fight other infections. So there'll be other infections that normally the body could take care of. It won't be able to, and that can make people very sick and even kill them. Because of HIV's high mutation rate, the shapes of its antigens change constantly. Since it takes about 14 days to develop an effective adaptive immune system to a new antigen, some populations of HIV are always able to stay ahead of the immune system. Populations of the virus with new antigens induce immune responses, but that takes time, and so they are able to spread before their host cells are killed. A healthy person has 500 to 1200 T helper cells per cubic millimeter of blood. For years, a person living with HIV may replace the T helper cells and appear healthy. But if left untreated, their T helper population will eventually decline. A person develops acquired immunodeficiency syndrome, or AIDS, when the number of T helper cells falls between 200 per cubic millimeter of blood. So here's what they should normally have, and here's the danger point for AIDS. A person without a healthy T helper population is vulnerable to many infections and sometimes of cancer. People with AIDS die from infections that don't harm healthy people. Condoms prevent transmission of virus particles between partners during sex. Virus particles do not stay intact outside the body. Limiting an individual's number of sexual partners will slow the spread of HIV in their community. Pre-exposure prophylaxis, or PrEP, is medication that can prevent initial HIV infections when taken every day. PrEP protects people who are at high risk of HIV infection by inhibiting reverse transcriptase, the enzyme that HIV needs in order to replicate. Taking antiretroviral drugs, or ART, makes HIV infection a manageable disease. ART is a mixture of drugs that prevent the replication of HIV and decreases the number of virus particles in the infected person. That allows their T helper cell population to remain stable. The ART drugs target HIV structures that are required for replication, and the virus does not tend to survive with mutations in these structures. Therefore, HIV cannot easily evolve resistance to the drugs. Without ART, the T helper cell population of a person living with HIV will eventually decline, leading to AIDS. However, most people live normal lifespans with HIV if they take ART consistently. Mother-to-child transmission of HIV is not well understood, but providing ART pills for the mother greatly reduces the risk of transfer to the child during pregnancy, childbirth, or breastfeeding.